Hello, welcome back to the continuation of the lecture on viruses, physiological psychology. This is the second of three videos. And to start, in order to understand the havoc that viruses wreak in our cells, um, it's important to have a good foundation in how protein synthesis works in cells. So proteins are absolutely vital for all sorts of processes inside of cells. Protein chemistry is basically what makes life work, what makes your cells function. Proteins are structural components of cells that hold things together. They are molecules that interact with the cell membrane in various ways to be receptors or to be recognition molecules that interact with other cells. They're enzymes that catalyze certain chemical reactions. So without proteins, life would cease to function. Your cells would cease to work. And in fact, any individual cell, like say a neuron versus a skin cell versus a liver cell versus a kidney cell, the reason those cells are different is because they have different proteins in them. So it's the proteins that make the cell do the things that the cell does. Um, and so it's super important. How does your body know what proteins it needs, or where do you get these proteins from? I mean, when you eat a hamburger, you know, okay, I'm getting a lot of protein here, but none of the proteins in that hamburger or in corn or whatever that you're eating, none of those proteins survive your stomach acids. Those proteins get broken down into components. One thing that makes all proteins what makes a protein, basically. A protein is typically a very large molecule which is formed out of smaller building blocks called amino acids. So here's kind of a diagram of a growing protein here, which is composed of several different amino acids, a yellow one, a blue one, a green one. This is a diagram that represents the fact that those are different amino acids. Our bodies use 20 different amino acids, and the ways that they combine and bind together will make unique proteins. A unique protein like insulin, for example, is going to be composed of hundreds of individual amino acids that are bound together by what are called peptide bonds. And because the amino acids are different, they're different compounds, leucine, isoleucine, I don't know. I don't know the names of all the amino acids. There's 20 of them. Um, they may have particular shapes and charges, and that will cause the protein to fold in various ways. And it's the shape of the protein typically that gives it its ability to either become part of a membrane, like a neurotransmitter receptor might be, or and certain enzymes might be membrane bound, or to float around in the cytoplasm of the cell, and to have a shape that might catalyze a chemical reaction or bind to a ligand or something like that. So these amino acids confer upon the protein a particular shape, and the shape typically confers upon its function. So when you eat a hamburger and you eat these proteins, these amino acids, these peptide bonds get broken down, the amino acids get absorbed, and then it's up to your cells to assemble the right amino acids in the right order to make insulin or to make a norepinephrine receptor or to make rhodopsin or something that your body is going to use for some kind of process. So the recipe for making that protein becomes very important. And that's basically what genes are. Genes are recipes for making proteins. So whenever a cell in your body needs to make a protein, the DNA, your genome gets consulted, and a process occurs whereby you go reading the recipe that's on the gene to manufacturing the protein with those amino acids in just the right order. And that process is summarized on this diagram here. More holistic view, your genetic information is stored on your DNA, but your DNA is in the nucleus of the cell, behind a membrane here, sort of a protected space that doesn't interact with the rest of the cell. And the DNA is a huge molecule. It's associated with lots of other molecules, and it never leaves the nucleus. So what happens is and the DNA molecule is huge. I mean, it's divided up into 23 chromosomes, but um, it's and those 23 chromosomes may contain thousands of different genes, thousands of different recipes for making proteins. I've seen, I don't know what the number is, 20,000, 30,000 genes, something like that. So um, you don't need to read the whole genome. You just, if you want to manufacture insulin, for example, you just need to find the insulin gene and you need to read the recipe for that gene. And that process is referred to as transcription. Transcription is the process of exposing one gene by partially unfolding the DNA to give enzymes and other molecules access to the information on that gene. And the gene is transcribed into a single strand of RNA called a messenger RNA molecule. So that process is referred to as transcription, where you take the information from one gene and you transcribe it onto a small RNA nucleic acid, which is small enough that it can leave the nucleus of the cell through a small pore or opening in the membrane of the nucleus. And then typically it directly enters what's called the endoplasmic reticulum, which is a large series of membranes surrounding the nucleus. In the neuron, it's in the soma of the cell, maybe a little bit of the dendrites of the cell. And it's here where the message on the messenger RNA molecule is translated into a protein. So on DNA, and therefore on the transcribed messenger RNA, there are base pairs. And every 3 base pairs is referred to as a codon, because those, depending on the identity of those base pairs, that will specify a particular amino acid. So there's another type of molecule called transfer RNA, which has 3 base pairs that are like the ones on the messenger RNA and are complementary to it. So you can see this G and this C are linking together, this U and this A, this G and this C are linking together. And these transfer RNAs, they have this recognition zone that briefly locks on to the messenger RNA if they're complementary to it. And on the other end of the molecule is carrying an amino acid. And peptide bonds are being formed between neighboring amino acids carried by tRNA molecules, so that as the messenger RNA moves its way through the reading frame of a ribosome, you add sequentially the amino acids that are next in line on the transcription of that gene. So the endoplasmic reticulum is this massive factory that requires certain concentrations of transfer RNA molecules with certain amino acids that you've gotten from your diet, from the hamburger that you've eaten, and your body can make some of them as well. And those get together and they form these proteins. And other stuff happens. The proteins are typically further moleculed, they're packaged in vesicles, they're shuttled off to different regions of the cell. So it's quite a production here. The physiology, the biochemistry of protein synthesis in cells is really quite amazing. As you can see, it involves lots of different components, and life wouldn't function without this process taking place. All right, so what's a virus? A virus is much simpler than a cell, and it is not a cell. It doesn't have a cell membrane. It doesn't have a lot of things that cells have. It's much, much simpler and much, much smaller than a cell is. It's basically a piece of DNA, or I should say it's basically a genome. Some viral genomes are DNA, but some viral genomes are RNA. That nucleic acid can either be double-stranded like it is in human cells and just generally in cells, throughout all species, or it can be RNA. And the genome is typically much smaller, rather than like our genomes contain around 20 to 30,000 genes and a lot of areas outside of genes, non-coding regions of DNA. So our DNA is a massive molecule. Viral genomes tend to be much smaller. They may contain 10 genes. Some viruses have larger genomes, could contain like 100 genes or something like that. Um, but but most viruses that you've heard about, they're going to have between 10 and 20 genes, and they typically, I don't know if they don't have any non-coding regions, but they don't have very. Most of their nucleic acid is devoted to coding regions of making 10, 12, 15 proteins, something like that. So they're very efficient genomes. They have a very small number of proteins that they make, and I shouldn't say that they make, that they code for, and that's basically it. And the DNA or RNA is usually not segmented into chromosomes. There might be a couple of different strands of these nucleic acids, or just maybe one long strand that's typically wound around like a helix, um, as shown in this diagram here. And then surrounding the nucleic acid is a shell, typically of proteins called a capsid. And then there is some larger shell around that, which is called the envelope. And sometimes there's a membrane, what looks like a cell membrane around that. So um, some viruses have a fatty envelope around it. Some viruses don't. Um, but as we'll see, if they do, it doesn't come from the virus itself. It's something they picked up along the way. So here you can see, this is diagram, it was just from Wikipedia, but the point of this diagram is that with just two proteins in this particular case, but if you make a lot of them, they'll get together in ways that create a geometric structure, like a icosahedron, like a 20-sided dice, if you've ever seen a 20-sided dice. And a lot of viruses seem to adopt this shape. They look almost spherical, so that the coronavirus that you see down on the screen basically has this shape here. Not all viruses have this shape, but it's a very common shape among viruses, and can be formed with a small number of proteins. Usually the envelope of a virus has more than two proteins, but it's not going to be a very large number. There will be other proteins that are sticking out of the envelope of the virus, which, if you look down at the coronavirus on the screen below me, you can see these red things that are sticking off the coronavirus. Those are proteins that this diagram here is calling an envelope protein. And those proteins are usually used to attach the virus to a cell that it's going to be infecting. So virus is a really simple thing. It needs some proteins to make the capsid, it needs some proteins to make the envelope, and it needs some proteins to recognize cells that it's going to infect, which are these envelope proteins. And that's typically what the majority of the genes in the viral genome do, is they make those proteins for making the nuclear shell, the viral shell, I don't know if shell's the right word, the capsid, the envelope, and these recognition protein molecules. Viruses sometimes will make other proteins as well, which are going to help the protein synthesis process, basically, or the genome copying process. So viruses, in order to survive, and they need to replicate, and so sometimes these proteins will help that replication process. And that's basically what a virus is. However, you'll notice that something that's not in those viruses, and that is there's no tRNA in those viruses. There's no amino acids in those viruses. There's no endoplasmic reticulum in those viruses. There's no room for any of that. So the virus has an interesting problem, which is it's got all this genetic information to make these proteins, to make more viruses, to look like itself, but it has no machinery for making that happen. And that's where infection comes in. So viruses are basically floating genomes with a protein shell that have no means to replicate themselves. And so they infect cells in order to steal the protein making machinery inside of those cells. That's why I opened this segment of the lecture with a review of protein synthesis, what all these parts of the cell are doing, because viruses need to get inside cells that have these things in order to basically steal them. And all of this machinery inside of a cell is being used to make all the proteins that this cell is going to need to survive. And the virus is going to step in and basically say, nope, you're going to use all this machinery to make more of me. So they're very nasty, nasty organism, and not really organisms, but they're horrible things. It's why computer viruses get the name viruses, right? Because they get inside the machinery of your computer and they use it to do the things that the computer virus wants to do, not the things that the user of the computer wants to do. These sorts of viruses get into your cells to make the cells do the things that the virus wants to do, not the things that the user of the cells want to do, which if you're sick, if you've got the cold or you've got the flu or you've got coronavirus, the reason you're sick is because the cells are not doing the things that you want your cells to do. They're doing the things that the virus wants them to do, which is basically replicate more virus, make those proteins that are needed, replicate the genome so that the virus will perpetuate itself. Okay. So viruses, you could think of them as parasites. Again, they're not living creatures, they're floating genomes, but they're basically taking over the transcription and translation making machinery of living cells in order to reproduce. And that process, partly because there's viruses in there, partly because you're not making your normal proteins, that will lead to dysfunction of the cells, sickness of the organism, and sometimes death of the cell, and sometimes death of the organism, depending on what cells are being affected and how many cells are being infected. So the lifespan or life cycle rather of a virus goes through several stages during the infection process. First, the virus must attach to a host cell. And typically that's, again, because the virus has got some proteins sticking out of it that recognize some proteins sticking out of the cell membrane of a cell in your body or bacteria or whatever. Viruses can infect anything. There are viruses for every creature to be infected by. And it's this process that allows for attachment. And it's also this specificity between the virus and the host cell. There's some kind of match that's occurring between them that determines the range of the virus. That is, what cells can the virus infect and what species can the virus infect. Viruses are exploiting something. Coronavirus, for example, the protein sticking out of the coronavirus attaches to some sort of angiotensin receptor, which is expressed in the lungs and is expressed in other places in the body, but seems to have an affinity for the human ACE receptor, as it's called. And because that ACE receptor is expressed in the lungs, we end up with a respiratory infection from coronavirus. Other viruses might have affinities for things sticking out of cells of the liver. I assume that's what hepatitis viruses do. Sometimes cells of the nervous system, like herpes viruses might have. Um, some viruses attack the GI tract, like polio virus. It just depends on the type of envelope protein that that particular virus makes and what sorts of how that matches up to certain types of cells that it infects. And of course, there's evolution that's occurring to make sure that these envelope proteins match better and better and better um, the types of cells that the virus does well in, replicates well in. Some viruses have very broad ranges. They may infect lots of cells in the body, or they may infect lots of different species. Other viruses have much more narrow ranges. I think the most common viruses on the planet, I think this is true, are called bacteriophages, and they only infect bacteria. And they only may infect particular species of bacteria as well. I read something recently that in a teaspoon of seawater, there's 10 million viruses or something, but they're almost all bacteriophages that don't affect humans. They only affect bacteria. That's why I say every living creature's got something that will infect it. And so this is a major target of the immune system. If the immune system can recognize the viruses based on their envelope proteins and attach things to the viruses that no longer allow it to interact with your cells, then the virus can't get inside the cells. And if it can't get inside the cells, it's not going to cause you problems. Once attachment takes place, the next thing that has to happen is the virus has to penetrate the host cell. And sometimes that occurs because the virus creates a pore in the membrane that the virus then goes through. Sometimes the cell itself envelops the virus, so the virus triggers an endocytosis type of response. Same thing that we talked about earlier when we talked about how neurons make synaptic vesicles, where the cell membrane surrounds the virus, and then the virus ends up inside the cell as a result. Some viruses may stay outside of the cell but inject their genome into the cell. That's how bacteriophages work. It's been proposed as a mechanism for viruses that infect multicellular organisms as well. I don't know, because I'm not a microbiologist or a virologist, whether or not that mechanism has been identified in any particular cell any particular virus that infects us. Once it's inside, then the virus will uncoat, which will allow it to release its genome into the intracellular space of the cell that it's infecting. RNA, if it's RNA, that will typically stay in the cytoplasm of the cell. If it's DNA, it will typically enter the nucleus of the cell. So it's got to have some mechanism for getting into the nucleus of the cell as well. Sometimes that DNA is incorporated into the genome of the host. Sometimes RNA is too. There are types of viruses that are called retroviruses, where the RNA is then back transcribed into, there's a term for it, I don't know what it is, back into DNA that often becomes incorporated into the host genome. And it is even possible then for that DNA to get perpetuated into the species if the host cell that incorporates that DNA doesn't die, and if it's a gamete that that can then pass that altered genome on. And that is referred to as horizontal gene transfer, where the genes from a virus, some bacteria can do this as well, where some of their genes end up getting incorporated into and then passed vertically down through progeny. I've been reading a few things about this. Apparently, sweet potatoes have evidence of bacterial DNA. There's a bacterium called agrobacterium, which is common in the soil, and we use it in laboratories to create genetically modified organisms, but it turns out nature genetically modifies organisms through horizontal gene transfer as well. And so some genes from these bacteria cause the roots of this plant to grow, and now we have the sweet potato, a relatively recent naturally genetically modified food as a result of this process. Um, but there are also other examples. Apparently, there was my sister-in-law just told me about a study that that um, proteins that allow the placenta to attach. Something about placental attachment. I don't know if it was to the womb or if it was to the fetus. Appear to be, I don't know if it was viral DNA or bacterial DNA. So this process might even contribute to evolution of species. In any event, back to the story of our viruses. Many of the viruses that you've heard about, their genomes are RNA, and sometimes very often they're single-stranded RNA that look just like messenger RNA, and so they can go straight to the ribosomes and the endoplasmic reticulum, and the viral proteins immediately begin to be translated from the genome of the virus. These will be the proteins that will form the capsid, the envelope proteins. They may also be enzymes that help to facilitate the other important thing that has to occur from the virus's perspective, which is its RNA has to get replicated, or DNA has to get replicated. So you have to make more and more copies of the viral genome if you want more and more copies of the virus. And sometimes the cell can recognize that there's foreign RNA here, and can get rid of it. That's another part of our immune response, but obviously that doesn't always happen. And so plenty of the messenger or the viral RNA gets replicated, and plenty of the viral proteins get translated, and then these proteins typically self-assemble around the genome of the replicated genome of the virus, and basically you're producing new viruses. And this is one of the reasons we get sick, is because your cells are devoting themselves now to making lots of copies of a virus rather than devoting themselves to doing the things that they normally do. And so one approach is to attempt to halt the virus being able to use the cellular machinery to make its own protein. And a number of antiviral drugs are aimed at that, are aimed at either halting viral genome replication or halting protein synthesis. But a lot of those drugs will also affect protein synthesis of normal cells. I mean, if you give a drug, it goes all over an antiviral drug, you swallow it, it goes all over your body. Only some of your cells are infected, and the antivirals are targeting those cells, but there's no way to get those drugs just into those cells. And so viruses are, I mean, I'm not a doctor either, so viruses, though, seem to me to be trickier than, say, a bacterial infection. Bacterial infections can be treated with antibiotics, and antibiotics, the doses will affect these tiny little bacteria but won't affect you. And so you basically can destroy the bacteria without significantly compromising the health of your normal cells. But with antiviral drugs, it's really difficult to. I think some of them are able to target certain things that the viruses are doing, but many of them just sort of broad spectrum block protein synthesis, and that's going to cause lots of side effects. So that can be tricky. But I know that antivirals are used in treating AIDS, for example. And then the final step of the process is that the virus has to be released. It doesn't do any good for the virus to stay inside your cell, making more and more copies of itself. And this is referred to as shedding the virus. So there's a few different mechanisms that occur. One mechanism is that your cell helps the virus the virus gets put into vesicles, basically, that, and there's sort of an exocytosis process where the vesicle fuses to the cell membrane. Sometimes the virus is able to retain some of that cell membrane, and that's how viruses get fatty membranes. Not many viruses, or I shouldn't say not many, I don't know. Some percentage of viruses have these fatty envelopes to them that look like cell membranes, and they're stolen basically from the cells that they've hijacked. Like everything else, these viruses just steal everything. And so some of them take this membrane with them, some of them don't. And that has some implications, I think, for how you might attempt to attack the viruses, whether they have these membranes in place or not. Sometimes the cells just rupture, which is referred to as lysis, as the cells become filled up with virus. Sometimes a cell detects that it's sick, basically, and that triggers a series of genes that cause a process referred to as apoptosis, where the cell kills itself from the inside, sometimes called cell suicide, which is a normal process that takes place in your body. When cells get sick, they sometimes undergo this apoptotic process. It's a cleaner way of dying than lysis. So with lysis, you're rupturing the membrane, stuff spilling into the extracellular space, and that can cause problems for neighboring cells. Apoptosis, the cell kind of chews itself up from the inside while it's sequestering portions of the cell, and so attempts to sort of cleanly destroy itself without having as much impact on the neighboring cells. And then again, exocytosis or budding can take place, which is often stealing cell membrane, which will usually trigger the cell to kill itself as well. So viruses very often will kill the cells that have manufactured them. And that obviously is another reason that we get sick, is because cells will die. And a virus could even become lethal if you're having some kind of organ failure as a result of lots of cells of a particular type being infected and dying. So I'm going to go back a slide here to make the point that when these viruses, however they find themselves getting released, they're now in the extracellular space, which likely puts them in contact with other cells. So this is how the virus will get worse. When you first get a cold or something, it's not so bad, but then it gets worse and worse and worse as more and more cells of a particular tissue type get infected, as these viruses now want to replicate themselves. But also, once the virus is released, it may end up in fluids that will allow the virus to be transmitted to other organisms or to the environment to later be picked up. Viruses, depending on where these cells are that are releasing them, can end up in blood, saliva, mucus, feces, basically any kinds of bodily fluids. And if those bodily fluids are getting out of your body, you're sneezing a lot, you're coughing a lot, you have diarrhea. There are certain types of nasty viruses that cause you to hemorrhage, to bleed. So there's lots of reasons why this release phenomenon not only will cause more of your cells to become infected, but will allow the virus to be contagious, allow you to be contagious, and for the virus to be transmitted to other members of the species. So that's basically the bad news about viruses. That's the way they work. They co-opt the cellular machinery inside your cells. They prevent your cells from doing what they normally do. They make more and more copies of themselves. The process of release typically destroys the host cell, allows these now to be more of the virus that can then more easily infect more cells. So you get this sort of rapid increase in severity of the infection in your body. And then population-wise, as more people become infected and more of these fluids for contagious viruses, for viruses that are contagious by bodily fluids, which, basically those that can lead to epidemics, pandemics, plagues, those sorts of things. are those types of viruses, then you get this, as more people get infected, more of them are around to infect other people. And so you get the kinds of um, epidemic curves that we've been seeing in the news these days. And so just on this very last slide, I'm going to end the second video after this slide. I just thought I would summarize some viruses. And even as I was putting these together, I realized there was a bunch of interesting viruses I left off the list. But I said, I'm just going to leave this to one table. So influenza and coronavirus are fairly similar in that their genomes are both single-stranded RNA. They're both about the same size, about 100 nanometers. The coronavirus has the shape which you see presented on the video of this icosahedral shape that's got all these pointy things sticking out of it. Influenza virus is more egg-shaped, although I guess there's a lot of diversity in influenza. There's several different strains for one thing. They have two proteins, the influenza viruses, one that starts with an H and one that starts with an N, that have been described. And that's why you hear like H1N1. That's the nomenclature of viruses that are related to flu, bird flu, Spanish flu, all these different types of flu. They tend to affect mammals, birds, which sort of host these viruses, and occasionally reinfect human populations. They affect mostly the throat, the upper respiratory tract, but obviously can have effects throughout our bodies as well. Coronavirus also targets the lungs, these ACE receptors, for example. That's a more lower respiratory problem, which can be, if you can't get oxygen into your system very easily, that's potentially going to cause system-wide problems. It may affect the GI tract as well of some species, including humans. Adenovirus is similar looking to the coronavirus. It's about 100 nanometers. It's also icosahedral, but it's got double-stranded DNA rather than single-stranded RNA, so it's different in that fact. It's another virus that tends to affect the upper respiratory tract. The common cold is an adenovirus, but there are a number of different adenoviruses that relate in severity from very mild sicknesses to very strong sicknesses. There's the adeno-associated virus, which is the virus that you read about in the methodology paper for today's class. And it is interesting in that it is non-pathogenic. It will infect cells, but it will not kill them. And so that's one of the reasons why it's been adapted for research purposes, and it's been talked about for gene therapy. It's also a DNA-based virus, single-stranded DNA in this case, which I presume makes it somewhat simpler to work with when you're using it for research purposes and attempting to look at the influence of certain genes. The problem with the adeno-associated virus is that it's tiny. It's only 20 nanometers, which means that if you want to use it in a research application to transfect an animal with some series of interesting genes, you're limited in the number of genes that you can pack into that tiny space. The larger the virus, the more genes it's going to have. The smaller the virus, the fewer genes it's going to have. One of the largest viruses, and obviously the largest one on this list, is the Ebola virus. It's got a flexible filament shape. It's basically a rod shape, and the the RNA is kind of wound around in this rod, but it has this sort of twisty shape. I think every Ebola virus adopts a slightly different shape. So it's long, flexible filament, about 8 times the size of the flu, which is also sort of rod shaped, but not a flexible rod shape. It infects a variety of tissue in humans, bats, and monkeys, and obviously is a very dangerous virus. HIV is 120 nanometers in size, is icosahedral as well, so it looks a bit like the coronavirus. It's based on single-stranded RNA, and some of the proteins that it makes allow that RNA to be back transcribed into DNA. So it's what's considered to be a retrovirus. Um, and it's a tricky virus for a number of different reasons that allows it to sort of avoid the immune system. Um, it infects humans and primates, immune systems, but also other parts of the body as well. Poliovirus is a little bit larger than the adeno-associated virus, but among the pathogens that affect humans, it is the smallest, and it affects the GI tract uh, most commonly of humans and primates, but by some mechanisms that I don't think are all that well understood, the poliovirus can work its way into the nervous system. And when it does that, that's when it has lethal effects. Poliovirus, of course, famously is one of the best studied viruses. And we have polio vaccines now that, for the most part, have eliminated polio in most parts of the world. Um, and so, hopefully, we'll have similar success with coronavirus. It's a somewhat tricky virus, but I think there's reason to believe that the vaccines can be developed. So in the third part of the lecture, we'll talk about research applications of viruses.